Hello everyone, my name is Carolina Sansaloni. I'm the Wheat Gene Bank Curator at CIMMYT, and today I'm going to share with you how we use DSI to analyze the structure, redundancy, and gaps in ex situ crop collection. As you know, a large proportion of valuable genetic variation is still hidden in the shelf of many gene banks, while of course the challenge for crop production becomes large and larger. At CIMMYT, we have two of the largest wheat and maize collection in the world. We conserve more than 29,000 accessions of maize and 140,000 accessions of wheat. But the first step is really to understand the collection, understand what we conserve for so many years. The ideal will be to phenotype the entire collection, but of course it will take a lot of time, it will be very expensive, and we have this interaction with the environment. But using genotyping technology, generating digital sequence information now become much more easy. With less time and of course a very reduced cost. And the main thing is we don't have any interaction with the environment. In this slide, you can see a global diversity analysis we perform using all DSI data. In this case, I'm sharing with you the wheat hexaploid accession. So each dot represents one accession of the collection. In total, we include 60,000 accessions. So in blue, you can see the land races collection. In red, the improved material from the breeding program. And in green, the synthetic material. We also perform a structure analysis to understand the collection. So as a conclusion, we see that diversity analysis stress role of key funding varieties in modern breeding programs, and also we identify land races with potential value in breeding programs. So there are a lot of opportunity to explore more the diversity conserved in the gene bank using this type of data. Another application of DSI is the curation of passport information. So all the data generated help us to understand, not only to understand the collection, but also to fix the passport information. In some cases, we found accessions that were registered as hexaploid, so 3 genomes, A, B, and D, and the others tetraploid, only A and B genome. But we found, looking at the genetic fingerprint, that in some cases, some accessions that are hexaploid in the passport data only has markers in the A and B genomes, means that are genetically tetraploid, and vice versa. So all this information allow us to fix the passport information, fix the taxonomy of the collection, and now we have more appropriate data to share with collaborators. Other uses of DSI is to check the redundancy of the collection, not only within collection, but across collection, across institutions collections. So we ran an analysis to check the redundancy of the CIMMYT gene bank, and we identify potential redundancy, and this helped us to manage more efficiently the collection. But also we check the redundancy across institutions, for example with ICARDA, and we could see the overlap of the collection or what unique material each institution has. Another important use is the gap analysis, so conservation gap analysis, that help to predict the distribution and quantify the current ex situ conservation status and give us an idea of potential future collection missions. So where to go and where to collect material that are not represented in the current collection. So summarizing, before digital sequence information, it was a big challenge to go into the gene bank and find something useful. As I mentioned before, at CIMMYT we have 140,000 accessions. So you imagine for a breeder or for a farmer or for any collaborator to find something useful, it was very, very difficult. Now, with the implementation of digital sequence information, it's much more easy. So we can narrow down the collection to a subset of useful material specific for a different challenge that a breeder or a farmer in different part of the world are facing. So it's much more useful and much more efficient searching in a gene bank using digital sequence information today. And to finalize, I would like to share a community of practice that we created between CIMMYT and CIAT. We brought together 33 institutions from 14 Latin American countries, and we trained them in the use of genomic and digital tools to enhance the conservation of genetic resources. So we characterized genetically several national gene banks, and now we are running analysis to understand the collection and to see unique material they conserve, overlap across gene bank, and also a comparison with CGIAR gene banks. And this is to not only to understand the collection, but also make more efficient the management or different national gene banks collection. With this, I would like to thank you for your attention and thank you very much. Goodbye.